Good day, ma'am. Good day, everyone. We are the Group 4 from BPED 4B, and my name is Kevin Pascual, your first reporter. And for today's video discussion, we were going to talk about Unit 9, which is gene therapy. But first, let me show you our learning objectives. First is to discuss the concept of gene therapy and its various forms. Second is to assess the benefits and detriments of gene therapy to human health. And third is to assess the issue's potential benefits and detriments to global health. Now, let me discuss what is gene therapy. So, gene therapy is a technique that modifies a person's genes to treat or cure disease. It is a technique that uses genetic material or a piece of DNA for a long-term treatment of genetic disorders. Gene therapy has provided treatments for incurable diseases that previously had only temporary remedies. Some of these diseases are partly or fully caused by genetic mutations such as cardiovascular disease, neurodegenerative disorders such as Parkinson's disease, vision disorders, blood clotting disorders such as hemophilia and diabetes mellitus. Gene therapy replaces a faulty gene or adds a new gene in attempt to cure disease or improve your body ability to fight disease. Gene therapy holds promises for treating a wide range of diseases such as cancer, cystic fibrosis, and heart disease. So here are the approaches to gene therapy. Gene therapies can work by several mechanisms. First, replacing a disease-causing gene with a healthy copy of the gene. Second is inactivating a disease-causing gene that is not functioning properly. And lastly is to introducing a new or modified gene into the body to help treat disease. Let's proceed to the types of gene therapy. Gene therapy are classified into two types. So first is the somatic gene therapy, and second is the germline gene therapy. First, let me discuss what is somatic gene therapy. In somatic gene therapy, the somatic cells of a patient are target for foreign gene transfer. In this case, the effects caused by the foreign gene is restricted to the individual patient only and not inherited by the patient's offspring or later generations. And also, somatic gene therapy is the ability to introduce genetic material or RNA into an appropriate cell type or tissue in vivo in such a way that it alters the cell's patterns of gene expressions to produce a therapeutic effect. Second is the germline gene therapy. In germline gene therapy, the functional genes which are integrated into the genomes are inserted in the germ cell, which is, or for example, sperm or eggs. Targeting of germ cells makes the therapy heritable. And also in germline gene therapy, when the DNA is transferred into the cells that produce reproductive cells, which is eggs or sperm in the body. So this type of therapy allows for the correction of disease causing gene variants that are certain to be passed down from generation to generation. So this is the end of my report. Again, my name is Kevin Pascual, and Elame Ponce will continue our report.
Good day, I'm Elamay P. Ponce from BPED 4B, and I am the second presenter of Group 4. And now, let's discuss the gene therapy strategies. So, letter A is gene augmentation therapy, or also known as GAT. In GAT, simple addition of functional alleles is used to treat inherited disorders caused by genetic deficiency of a gene product. Example of gene augmentation therapy, or also known as GAT, has been applied to autosomal recessive disorders. Dominantly, inherited disorders are much less amenable to GAT. So these strategies is used to treat diseases caused by a mutation that stops a gene from producing a functioning product such as protein. The delivery of a correct copy of the gene is expected to restore the production of the defective or missing protein and thus revert the disease phenotype. This type of a gene augmentation therapy is often also referred to as a gene replacement therapy. So in other words, when we say gene augmentation therapy, it aims at compensating a pathological mutation or improving the homeostasis of a compromised cell that can be used to resolve recessive loss of function disorders. The gene augmentation therapies are being undergone for a small number of recessive single gene diseases. This is only successful if the effects of the disease are reversible or have not resulted in lasting damage to the body. For example, this can be used to treat loss of function disorders such as the cystic fibrosis by introducing a functional copy of the gene to correct the disease. And the second one of gene therapy strategies is the letter B, which is targeted killing of specific cells. It involves utilizing genes encoding toxic compounds or suicide genes or prodrugs, reagents with conferred sensitivity to subsequent treatment with a drug to kill the transfected or transformed cells. This general approach is popular in cancer gene therapies. The target killing of specific cells can be done by two ways. First is the direct cell killing is possible if the added genes create a lethal toxin or suicide genes that produce a highly toxic product which kills the cells. Second one is the indirect killing or prodrug gene is inserted conferring vulnerability to killing by a later administered medication. The prodrugs is involves activation of particular enzyme specifically in cancer cells which convert a non-toxic product. Immunostimulatory genes are used in indirect cell killing to stimulate or amplify an immune response against the target cell. The inserted DNA causes expression of a protein that marks the cells so that the diseased cells are attacked by the body's natural immune system. It is essential with this method that the inserted DNA is targeted appropriately to avoid the death of cells that are functioning normally. And to continue our discussion, here is Kevin Mendoza.
Hi everyone, I'm Kevin Mendoza from BPED 4B. So to continue our lesson in gene therapy strategies, I will be going to discuss the targeted inhibition of gene expression and also the targeted gene mutation correction. So first, I will be going to discuss the targeted inhibition of gene expression. So this is to block the expression of any disease gene or a new gene expressing a protein which is harmful for a cell. This is particularly suitable for treating infectious diseases and some careers. So as we can see in this figure in targeted inhibition of gene expression, we have the antisense TFO or ODN or the antisense genes, which is the disease cells containing mutant or harmful disease. So through that inhibition, we have the DNA and RNA triplex and also the RNA or the DNA and RNA duplex. So through that, we have the transcription block, which is to block the expression of pathogenic gene. So in this figure is to inhibit the target gene expression in disease cell that antisense mRNA coding gene inserted vector or triplex forming oligonucleotides or TFO or antisense oligonucleotides or ODN can be introduced, which will inhibit the gene or the gene expression either by forming DNA or RNA triplex inside the nucleus or forming. So next we have the targeted gene mutation correction. So it is used to correct the defective gene to restore its function, which can be done at genetic level by homologous recombination or at mRNA level by using therapeutic ribozymes or therapeutic RNA editing. So as we can see in this figure, which is the targeted gene mutation correction, we have the disease cell or the mutant gene X, and also in the middle we have the corrected gene, and in the other side we have the normal phenotype or the genetic mutation corrected to restore functional gene. So in this figure, this is used for disease caused by mutation. So the corrected gene will be swapped by the mutant gene X, and then the disease or the disease cells will become normal after the correction of mutation by gene therapy. So to continue our report, here is John Ramos to discuss the method of gene therapy.
Good day everyone, my name is John Ramos Ramos from BPED 4B. I am the fourth reporter of Group 4, Methods of Gene Therapy. Primary goal of gene therapy is to determine whether a new gene is capable to replace or inactivate a mutated gene in order to treat or help the body to fight a certain disease. And in order for the gene to be distributed to the cells, The process is called transportation. At this process, a transporter called vector takes place. It is genetically engineered to deliver the gene. A vector is made of altered virus. Retroviruses are among the most widely used viral vectors in gene therapy. Certain viruses are also used as vectors because they can deliver the new gene by infecting the cell. Vectors can be given intravenously. Example of this is injection in the vein and specific tissue in the body. The latest is the surgery in which vectors were delivered into a specific area of the body. There are mainly two approaches for the transfer of genes in gene therapy. These are ex vivo and in vivo, which are gonna be reported by Miss Cherry Gil Peralta and Miss Risa Negrido.
Good day everyone, I am Cherigil V. Peralta from BPED 4B, and for today I will be going to discuss ex vivo gene therapy. In this mode of gene therapy, genes are transferred to the cell grown in culture, transformed cells are selected and multiplied and then introduced into the patient. The use of autologous cell prevents the cell from being rejected by the immune system. The cells are taken from the patient and then it will be treated and cultivated in culture before being reintroduced back into the same body. This method may be used in tissues such as hematopoietic cells and skin cells, which can be withdrawn from the body. It will be genetically repaired outside and then reintroduced into the patient's body where they engrafted and live for a long time. Example of ex vivo gene therapy is treating conditions that affect the blood, such as hemophilia, which are person's own self-renewing stem cells can be engineered with functional working gene and then delivered into their body. Now, to continue the discussion, Ms. Riza Negrido will be discussing about the vivo gene therapy.
Thank you, Miss Cherigale Peralta. So, next is in vivo gene therapy. So, in vivo method of gene transfers involves the transfer of cloned genes directly into the tissues of the patient. This is done in case of tissues whose individual cells cannot be cultured vitro in sufficient numbers, like brain cells, and or where reimplementation of the cultured cells in the patients is not efficient. So liposomes and certain viral vectors are employed for this purpose because of the lack of any other mode of selection. In case of viral vectors, such type of cultured cells were often used which have been infected with the recombinant retrovirus in vitro to produce modified viral vectors regularly. These cultured cells will be called as vector-producing cells or VPCS. The VPCS transfer the gene to surrounding disease cells. The efficiency of gene transfer and expression determines the success of this approach because of the lack of any way for selection and amplification of cells which take up and express the foreign gene. So this gene therapy is produced of cells like transfer in one person surrounding disease of the cells. Then we have therapeutic genes are inserted into viral DNA, lysosome, or as plasmid or DNA. We have a viral vector, liposome, plasmid, or DNA. Number two is genetically altered DNA is inserted into patient's body by direct tissue injection. So need to inserted in other patients directly. Number 3. The inserted DNA is incorporated into the specific cell and produce the needed protein encoded by the inserted gene. So to continue the discussion, here is Miss Arian Joyce Pineda.
Good day. I'm Arian Joyce S. Pineda from BPED 4B, and I'm here to discuss the target sites of gene therapy. Target sites for gene therapy. Therapeutic genes have to be delivered to specific target sites for a specific types of disease. Target cells for gene transfer. So the first disease is cancer, and the target cells are tumor cells, antigen-presenting cells, blood progenitor cells, T cells, fibroblasts, and muscle cells. So cancer develops when the body's normal control mechanism stop working. Old cells do not die and instead grow out of control, forming new abnormal cells. These extra cells may form a mass of tissue that's called a tumor. Second is inherited monogenic disease, and the target cells are lung endothelial cells, macrophages, T cells, blood progenitor cells, hepatocytes, and muscle cells. So, monogenic disorders or disease are caused by variation in a single gene and are typically recognized by their striking familial inheritance patterns. Examples include sickle cell anemia, cystic fibrosis, and Huntington disease. So next is infectious disease, and the target cells are T cells, blood progenitor cells, antigen-presenting cells, and muscle cells. Infectious disease are disorders caused by organisms such as bacteria, viruses, fungi, or parasites. Many organisms live in our bodies. They are normally harmless or even helpful, but under certain conditions, some organisms may cause disease. Some infectious disease can be passed from person to person. Next is cardiovascular disease. The target cells are endothelial cells and muscle cells. Cardiovascular disease or CBD is a general term for conditions affecting the heart or blood vessels. It's usually associated with the buildup of fatty deposits inside the arteries and an increased risk of blood clots. So some examples of cardiovascular disease is heart failure and stroke. Next is rheumatoid arthritis. The target cells is synovial lining cells. Rheumatoid arthritis or RA is an autoimmune and inflammatory disease, which means that your immune system attacks healthy cells in your body by mistake, causing inflammation in the affected parts of the body. RA or rheumatoid arthritis mainly attack the joints. And lastly, cubital tunnel syndrome. The target cells is nerve cells. So when we say cubital tunnel syndromes, it happens when the ulnar nerve, which passes through the cubital tunnel on the inside of the elbow, is injured and becomes swollen and irritated. So to continue the discussion, the next reporter is Miss Ruela Ann Prisima.
Good day everyone, my name is Rueda Anpursima from BPED 4B. Today I'm going to report the advantages and disadvantages of gene therapy. So let's start in the advantages of using gene therapy. So number 1 is gene silencing. In the case of an HIV-infected person, gene therapy and gene silencing can be protect the patient from the pain and suffering before the disease progresses. So means the regulation of gene expression in a cell to prevent the expression of the certain gene. So next is gene therapy is potential used to eliminate and prevent hereditary diseases such as cystic fibrosis. It can also a potential way to treat heart disease, AIDS, and cancer. So means the technique that modifies a person's genes to treat or cure disease. And now let's proceed the disadvantages of gene therapy. The novelty of gene therapy methods is one advantage. Stimulation of immune response. The gene injected by a virus may cause immune responses due to the presence of the virus inside the body and the pathogenic potential of viral vectors. So the example is unwanted immune system reaction. Your body's immune systems may see the newly introduced the viruses and intruders and attack them. This may cause inflammation of serious cases, organ failures. So last is the generation of genetic disorders due to the presence of multigene. The genetic materials transferred may not necessarily enter the target cell. Even if it does, it may not be placed an appropriate place in the genome. So that's the end of our discussion. Thank you.